Good afternoon, everyone. I am Alexandre BELOT, Professor of Pediatrics here in Lyon and clinical scientist at Hospices Civil de Lyon and Syrie. Now, I'm here to talk to you about genomic signature in lupus patients for personalized therapy. So, as you know, lupus is the paradigm of the autoimmunity and represents a debilitating disease. It's also the autoimmune disease with the most important genetic contribution. To date, lupus has not been able to benefit from the new drug discovery, contrary to other autoimmune diseases such as rheumatoid arthritis, psoriasis, vasculitis. This is probably due to its extreme heterogeneity and how to clearly define a lupus. So what is a lupus? Except for belimumab, there is no FDA-approved drug for lupus since 50 years, and almost all classical clinical trials have failed for lupus. Thus, companies are even reluctant to proceed with new trials. So according to all experts around the world, it's still an enigma. Why lupus does so badly in clinical trials? This is, I feel, the time to enter in a new era in the management of autoimmune disease, as in cancer biology, and we felt that our expertise in next-generation sequencing could be a solution. We focus on pediatric onset SLE at first because it's most severe. There is the highest contribution of genetics and a low level of confounding factors such as obesity, tobacco use, other treatments. The technique we use is next-generation sequencing by panel for whole genome sequencing to detect the individual genetic code of patients and select rare and predicted pathogenic variants. We have applied this before in a large panel of 117 lupus patients, and we have created a specific genetic algorithm to identify the genetic code of each patient. We can identify the susceptibility in a single patient relying on monogenic defect, oligogenic defect, digenic defect. We can actually highlight the genetic susceptibility in a single patient that enable to have targeted and personalized treatment to suit their genetic code. Our objective is to dissect the individual genetic contribution in lupus patient and to define an immunocard associating the impaired immune response to the genetic defect. We will create a database of this precious information to create targeted treatments. We will use the genome exome sequencing strategy in juvenile lupus together with fast immune functional assay. And actually, we have already this assay, such as the interference signature in our routine lab here in Lyon, and we are working on nucleic acid sensing assay as well. We have already been successful with this strategy, and together with Frédéric Rioloca's group in Imagine, we have identified that SOX1 haploinsufficiency is associated to various autoimmune conditions, including lupus. So what is SOX1? SOX1 is inducible suppressor of cytokine signaling, the most potent. It's a natural JAK inhibitor we all have in our cells. And we successfully treated our patients with lupus with a JAK inhibitor that complemented the immune genetic defect, restoring normal cytokine signaling. We have actually identified a total of 10 patients from 5 countries with various autoimmune diseases associated to SOX1 defect. Behind this study is myself, a clinician scientist dedicated to rare disease. I'm head of RAISE Lyon, the National Reference Center of Rare Rheumatic and Autoimmune Disease in Children, and a co-chair of the network FEDESER with Eric Achula, a large connection of clinician experts in inflammatory disease. Today, we have identified several genetic causes of lupus in the lab in the series Lyon. We have performed transcriptomic data analysis for this specific patient, and we are still looking for specific biomarkers in order to tailor therapy in juvenile lupus. Using CRISPR-Cas9 strategy, we have also generated mice preclinical models carrying mutation of genes that are involved in monogenic lupus in order to test drugs on them. We have a nice experience of joint project with pharmas in the US, namely EMD, Serono, and BI. Now, we are looking for funding and partners to build together the immunogenetic card of juvenile lupus, adult lupus, and create personalized therapy for them rather than large-scale clinical trials that have failed in the past. Once we will have treated the 25,000 patients in France, we can maybe apply to the whole lupus community around the world and move to other systemic autoimmune disease that may share the same concept of ideas that the disease is driven by genomics. There are different perspectives, such as joint project for drug testing or the current RHU call that I'm sure we can build together a new era for the treatment of lupus patients. Thank you very much.